So here's something kind of wonderful to think about as you're lying there tonight. You are, right now, in this exact moment, doing the very thing that has confused the smartest scientists on the planet for centuries. You're getting ready to fall asleep, and nobody, not the Nobel laureates, not the neurologists, not the people running sleep labs with millions of dollars of equipment, nobody fully understands why you need to do this. Not completely. Not in a way that ties it all together into one neat explanation. And I think that's kind of beautiful, honestly. Because sleep is this thing we all do, every single night, this enormous chunk of our lives, roughly a third of it, if you're lucky. And for most of human history, we had almost no idea what was actually happening during those hours. We just knew we needed it. We knew that if we didn't get it, things started to fall apart pretty quickly. But the why and the how, those were mysteries. Deep, stubborn mysteries. And a lot of them still are. So tonight, let's just wander through this together. Let's talk about what we know, what we figured out, what we got wrong for a really long time, and what's still genuinely puzzling. And if you drift off somewhere along the way, well, that's kind of the point, isn't it? You'll be doing the very thing we're talking about. Let's start way back. And I mean way back. Because humans have been trying to explain sleep for as long as we've been trying to explain anything. The ancient Greeks had some ideas. And like a lot of ancient Greek ideas, they were creative and confident, and mostly wrong, but in interesting ways. Aristotle, and this is around 350 BC, he had this theory that sleep was caused by warm vapors rising from your stomach after you ate a meal. The idea was that digestion produced these fumes, and they traveled up to your brain, and the brain cooled them down, and that cooling process somehow made you unconscious. It's a beautiful theory in a way. It has this lovely internal logic. You eat dinner, you feel sleepy. Therefore, eating must cause sleeping. And if you think about it, he wasn't totally crazy. We do get drowsy after big meals. There's a real physiological reason for that, which we'll get to later. But the mechanism he proposed, the warm vapors and the cooling, that's not what's happening at all. What's interesting, though, is that Aristotle was already asking the right questions. He wrote a whole treatise called On Sleep and Sleeplessness, and in it, he's wrestling with things like, is sleep a property of the body or the soul? Why do all animals seem to need it? What's the difference between sleep and just being unconscious? These are genuinely hard questions, and some of them took us more than 2,000 years to even begin answering properly. Now, for a very long time after the Greeks, the dominant view was basically passive. People thought of sleep as an absence. You were awake, and then your brain just turned off, like a candle going out. Sleep was nothing. It was the gap between the interesting parts. And this wasn't just folk wisdom. Serious scientists held this view well into the 20th century. The assumption was that your brain was essentially inactive during sleep, just idling, waiting for morning. And this assumption shaped how people thought about sleep for centuries. If sleep is nothing, then there's not much to study, is there? You can't study nothing. But here's the thing. Even before we had the tools to look inside a sleeping brain, there were clues that this passive view was wrong. Obvious clues, really, if you stopped to think about them. Dreams, for one. If your brain is just switched off, where are the dreams coming from? Why are they so vivid, so emotionally intense, so narratively complex? A brain that's doing nothing doesn't generate elaborate stories about showing up to work in your pajamas or flying over cities you've never visited. Something is clearly happening in there. And then there were the clinical observations. Doctors had noticed for centuries that sleep deprivation did terrible things to people. Not just made them tired, it made them confused, paranoid, emotionally unstable, physically sick. In the 1890s, a Russian physician named Marie de Menacin did some of the earliest experimental work on sleep deprivation using puppies, which is heartbreaking, but was considered acceptable science at the time. She kept them continuously awake, and they all died within a few days. Every single one. And when she examined their brains afterward, she found significant damage, hemorrhages, degeneration of brain cells. This was striking. Whatever sleep was, it wasn't optional. It wasn't a luxury. It was, somehow, keeping the brain alive. But even with results like that, the scientific mainstream didn't really take sleep seriously as a field of study for decades. It was considered a backwater. If you were an ambitious young neuroscientist in the early 20th century, you studied waking consciousness, you studied reflexes, you studied learning and memory. You didn't study sleep. Sleep was boring. Sleep was the absence of the interesting stuff. And then, in 1929, something happened that changed everything, even though almost nobody realized it at the time. A German psychiatrist named Hans Berger published a paper announcing that he had recorded electrical activity from the human brain. He called his invention the electroencephalogram, the EEG. And this is one of those moments in science where a new tool doesn't just help answer existing questions. It reveals that you've been asking the wrong questions entirely. Berger was a fascinating character, by the way. He'd been obsessed with the idea of psychic energy since he was a young man. There's this story, and it's one he told himself, so take it with a grain of salt, that when he was serving in the German military as a young man, he fell from his horse during an exercise and nearly got crushed by a horse-drawn cannon. That same day, his sister, who was miles away, had a sudden overwhelming feeling that something terrible had happened to him, and she convinced their father to send a telegram asking if Hans was all right. Berger became convinced that his brain had transmitted some kind of energy at the moment of crisis, and his sister had received it. He spent the rest of his career trying to find that energy. He never found psychic transmission, of course, but what he found instead was arguably more important. He found that the brain produces continuous, measurable electrical rhythms, waves, patterns, and those patterns change depending on what the brain is doing. Now, Berger's work was initially met with a lot of skepticism. People thought his recordings were artifacts, just electrical noise from muscles in the scalp, not actual brain activity. It took several years and independent confirmation from other researchers, particularly Edgar Adrian and Brian Matthews in Cambridge, England, in 1934, before the scientific community started to accept that yes, the brain really does produce these electrical rhythms, and yes, you really can record them from the surface of the skull. But here's where it gets relevant to our story. Once people started using EEGs on sleeping subjects, the passive theory of sleep collapsed almost immediately, because a sleeping brain doesn't look quiet on an EEG. It doesn't look like a brain that's turned off. It looks like a brain that's doing something completely different from waking, but doing it intensely. The patterns change. The waves get bigger, slower, more synchronized, and they keep changing throughout the night, cycling through distinct stages in a pattern that repeats roughly every 90 minutes. This was a revelation. Sleep wasn't one thing. It wasn't an off switch. It was a sequence of different brain states, each with its own characteristic electrical signature, cycling over and over throughout the night. And nobody had known this. For all of human history, nobody had known this. You'd go to bed, you'd wake up in the morning, and you had no idea that your brain had been cycling through this elaborate choreography of different states all night long. The person who really cracked this open was a graduate student at the University of Chicago named Eugene Oserinsky. And his story is one of those wonderful tales of accidental discovery that science is full of. It was 1951, and Oserinsky was working in the lab of Nathaniel Kleitman, who was one of the very few people at that time who took sleep research seriously. Kleitman had been studying sleep since the 1920s. He'd even spent 32 days living in Mammoth Cave in Kentucky in 1938, trying to understand what happened to sleep patterns when you removed all exposure to daylight. He was dedicated, to say the least. Oserinsky was a struggling graduate student. He'd already failed out of one program, he had a family to support, and he was working with old, temperamental EEG equipment that kept breaking down. Kleitman gave him a project that was frankly considered a bit of a dead end, watching the eye movements of sleeping infants. The idea was just to track when babies' eyes moved during sleep and see if there was any pattern. And Oserinsky noticed something strange. At certain points during sleep, the baby's eyes would start moving rapidly back and forth under their closed eyelids. Really rapidly, almost like they were watching something. And when he looked at the EEG recordings during these periods, the brain activity looked completely different from the rest of sleep. It looked almost like the brain was awake, fast, low-voltage, desynchronized waves, the kind of pattern you see in an alert, active brain. But the babies were clearly asleep, deeply asleep, actually. Their muscles were relaxed. They were hard to wake up. Oserinsky brought this to Kleitman, and they decided to investigate further, this time with adult subjects. They confirmed it. There were periods during the night, multiple periods, recurring in a regular cycle, when the eyes moved rapidly, the brain lit up with wake-like activity, and the sleeper was actually harder to rouse than during other stages of sleep. And here's the kicker. When they woke people up during these periods, the subjects almost always reported vivid dreams. When they woke them during other stages, dreams were rare or vague. They discovered REM sleep, rapid eye movement sleep, and with that discovery, they'd split the night into two fundamentally different kinds of sleep, REM and non-REM, and opened up an entirely new field of science. Oserinsky and Kleitman published their findings in 1953 in the journal Science. And it's not an exaggeration to say that this paper is the foundation of modern sleep research. Everything we know about sleep stages, sleep cycles, the relationship between sleep and dreaming, the architecture of a night's sleep, all of it traces back to that moment when a broke graduate student noticed something odd about the way sleeping babies move their eyes. And what makes this discovery so compelling is what it implied. If the brain is highly active during REM sleep, burning almost as much energy as it does when you're wide awake, then sleep clearly isn't rest in the way people had always assumed. Or at least, not all of sleep is rest. Something is happening during REM that the brain needs to do. Something important enough that it dedicates roughly 20 to 25% of your total sleep time to it every single night, in cycles that get longer as the night goes on. But REM was only half the picture, because the rest of the night, the non-REM portion, turned out to be just as interesting, just in a completely different way. When researchers started looking more carefully at non-REM sleep with better EEG equipment, they found that it wasn't just one uniform state either. It had its own stages, its own progression, its own characteristic brain waves. And the deepest stage of non-REM sleep, what we now call slow wave sleep or deep sleep, produced these enormous rolling electrical waves that swept across the entire brain in synchronized pulses. Imagine the difference between a choppy sea with waves going every direction. That's your waking brain, lots of different areas doing lots of different things. And a calm ocean with these huge, slow, powerful swells all moving together. That's deep sleep. Billions of neurons firing in unison, then going quiet, then firing again, in a slow, majestic rhythm about once per second. And nobody knew what those waves were for. They still don't know entirely. But we've learned a lot since the 1950s, and some of what we've learned is genuinely surprising. Because it turns out that those slow waves aren't just an interesting electrical pattern. They seem to be doing something functional. Something the brain can't do while you're awake. Something that requires you to be offline, disconnected from the outside world, essentially unconscious. And the leading theories about what that something is, well, they involve everything from memory consolidation to literal brain cleaning, and they're some of the most fascinating ideas in all of neuroscience. But to understand why those theories matter, and why they took so long to develop, we need to talk about what happens to your body when you fall asleep. Not just your brain, your whole body. Because sleep isn't just a brain phenomenon. Your heart rate changes, your temperature drops, your hormones shift, your muscles do something really strange during REM that we haven't even gotten to yet. And all of these changes happen on a schedule, orchestrated by a system so elegant and so ancient that it predates not just humans, not just mammals, but possibly even the evolution of brains as we know them. Fruit flies sleep. Jellyfish sleep. And jellyfish don't even have a central nervous system. Whatever sleep is doing, it's so fundamentally important that evolution has preserved it across hundreds of millions of years, even in creatures that pay an enormous survival cost for it. Think about that. Sleep makes you vulnerable. You can't eat, you can't watch for predators, you can't reproduce. Every hour you spend asleep is an hour you could be doing something that directly helps you survive and pass on your genes. And yet, every animal we've ever studied does it anyway. That tells you something. That tells you sleep isn't optional. It isn't a luxury. It's doing something so critical that the cost of not doing it is worse than lying unconscious and defenseless for hours at a time. So let's walk through what actually happens when you fall asleep tonight. And I mean the whole sequence, step by step, because it's more orchestrated than you might expect. It starts before you even feel drowsy. Hours before, actually. There's a tiny cluster of neurons sitting right above the point where your optic nerves cross. It's called the suprachiasmatic nucleus, which is a mouthful, so most people just call it the SCN. It's about the size of a grain of rice, maybe 20,000 neurons, and it's the master clock of your entire body. This little cluster receives direct input from special light-sensitive cells in your retinas, not the rods and cones you use for vision, but a separate population of cells discovered only in the early 2000s, cells containing a photopigment called melanopsin that's particularly sensitive to blue light. And these cells don't help you see the world. Their job is simpler and in some ways more important. They tell your brain what time it is. When those melanopsin cells detect bright, blue-rich light, the kind you get from sunlight, or, as it turns out, from phone and computer screens, they send a signal to the SCN that says, essentially, it's daytime. And the SCN uses that information to coordinate a cascade of biological events throughout your entire body. It's not just one signal. The SCN influences dozens of downstream processes: body temperature, cortisol release, digestion, cell division, immune function, all of it timed, all of it rhythmic, all of it cycling roughly once every 24 hours. This is your circadian rhythm, from the Latin circa diem, meaning about a day. And here's what's clever about it. The SCN doesn't just respond to light. It generates its own rhythm internally. If you put a person in a cave with no light cues at all, and people have actually done this, there's a famous experiment from 1962 where a French geologist named Michel Siffre spent 2 months in an underground cave in the Alps with no clock, no sunlight, no way to tell what time it was. What happens is that the person keeps sleeping and waking on a cycle, but the cycle drifts. It's not exactly 24 hours. It's usually a little longer, somewhere around 24 hours and 10 to 15 minutes, which means the SCN has its own built-in oscillation, its own sense of time, and the light coming through your eyes each day just nudges it, resets it, keeps it synchronized with the actual rotation of the Earth. Without that daily reset, you'd slowly drift out of phase with the world around you, which is essentially what happens with jet lag. Your SCN is still running on the old time zone, and it takes a few days of light exposure in the new location to pull it back into alignment. So as evening approaches and the light fades, the SCN does something important. It sends a signal through a surprisingly roundabout neural pathway to a tiny gland buried deep in the center of your brain called the pineal gland. And the pineal gland, in response, starts producing melatonin. Now, melatonin is one of those words that's entered popular culture, and a lot of people think of it as a sleep drug, something that knocks you out. But that's not really what it does. Melatonin is more like a messenger. It's a signal of darkness. It doesn't force you to sleep so much as it tells your body and brain that nighttime has arrived and it's an appropriate time to sleep. It opens the gate, so to speak. The actual process of falling asleep involves a different set of mechanisms. And this is where it gets interesting, because there's a second system working alongside your circadian rhythm, and it operates on completely different principles. It's called sleep pressure, or more technically, the homeostatic sleep drive. Here's how it works. From the moment you wake up in the morning, a chemical called adenosine starts accumulating in your brain. Adenosine is essentially a byproduct of neural activity. When your neurons burn energy, adenosine is one of the things left over. And as it builds up over the course of the day, it binds to specific receptors in your brain, and the effect of that binding is to make you feel progressively sleepier. The longer you've been awake, the more adenosine has accumulated, and the stronger the pressure to sleep becomes. Now, you might already be thinking about caffeine, and you'd be right to. Caffeine works by blocking adenosine receptors. It doesn't reduce the amount of adenosine in your brain. The adenosine is still building up. But caffeine molecules are shaped just enough like adenosine that they can sit in the same receptors without activating them. It's like putting a broken key in a lock. The real key, adenosine, can't get in, so your brain doesn't get the sleepiness signal. But the adenosine is still accumulating behind the scenes, which is why, when the caffeine eventually wears off and gets metabolized by your liver, all that pent-up adenosine suddenly floods into the now unblocked receptors, and you crash. That afternoon, coffee didn't eliminate your sleep pressure. It just hit it for a few hours. So you've got these two systems. The circadian rhythm saying, it's nighttime, sleep is appropriate now, and the homeostatic drive saying, you've been awake for 16 hours, you need to sleep. And when they align, when both signals converge, that's when you feel that irresistible wave of drowsiness, that heavy eyelid, can't keep your head up feeling. It's two independent biological systems agreeing that right now is the time. And then you actually fall asleep. And what happens next is a sequence so precisely choreographed that it unfolds almost identically every night of your life. Your brain doesn't just switch off. It transitions through stages, and the order and timing of those stages follows a pattern that sleep researchers call sleep architecture. In the first cycle of the night, you descend relatively quickly through the lighter stages of non-REM sleep. Stage 1, which is that drowsy, drifting, half-aware state where you might have those little hypnic jerks, those sudden muscle twitches that feel like you're falling. And then into stage 2, where your brain starts producing these distinctive electrical signatures called sleep spindles and K-complexes. Sleep spindles are these beautiful little bursts of activity, rapid oscillations that last about half a second to a second, and they seem to play a role in protecting your sleep from external disturbances. When a noise happens in your environment during stage 2 sleep, your brain often responds with a K-complex, a sharp, high-amplitude wave, followed by a spindle, and the effect seems to be a kind of neural gating, a way of saying, noted, not important, stay asleep. It's your brain actively defending your unconsciousness, and then you sink deeper into slow-wave sleep. That deep stage we talked about earlier with those massive synchronized neural oscillations. And in the first cycle of the night, this deep sleep phase is long, maybe 30 or 40 minutes. Your brain is producing these enormous delta waves. Your muscles are relaxed but not paralyzed. Your heart rate has dropped. Your blood pressure has fallen. Your core body temperature has decreased by nearly a full degree Celsius. Growth hormone is surging. In fact, the majority of your daily growth hormone release happens during this first bout of deep sleep, which is one reason why sleep is so critical for children and why athletes who don't sleep well recover more slowly from training. Your immune system is ramping up certain activities. Cytokines, signaling proteins involved in immune response, are being released in higher quantities. This is part of why you feel so sleepy when you're sick. Your body is essentially demanding more deep sleep because it needs the immune boost that comes with it. After maybe 70 to 90 minutes total, you complete the first cycle by rising back up through lighter sleep and entering your first REM period. And here's where something genuinely strange happens. Your brain, which was just producing these slow, synchronized waves, suddenly shifts into a pattern of activity that looks almost indistinguishable from wakefulness. Fast, desynchronized, chaotic electrical activity. Your eyes start darting back and forth beneath your closed lids. That's the rapid eye movement that gives this stage its name. Your breathing becomes irregular. Your heart rate fluctuates. And your brain does something remarkable, and when you think about it, a little unsettling. It paralyzes you. Not your diaphragm, you keep breathing, and not your eye muscles, they're still moving. But virtually every other voluntary muscle in your body is actively inhibited. The signal comes from a region in the brainstem called the sublaterodorsal nucleus, and it works by releasing neurotransmitters, glycine and GABA primarily, that hyperpolarize motor neurons in your spinal cord, essentially making it impossible for the movement commands your dreaming brain is generating to reach your actual muscles. You are, for all practical purposes, temporarily paralyzed from the neck down. And this is not a malfunction. This is a feature. Because during REM sleep, your brain is generating vivid, complex, often physically active dream scenarios. You're running, flying, fighting, falling. If those motor commands actually reached your muscles, you'd be acting out your dreams. You'd be thrashing around, getting out of bed, potentially hurting yourself or anyone sleeping next to you. And we know this because there's a condition, REM sleep behavior disorder, where this paralysis mechanism fails. People with this condition do act out their dreams. They punch, kick, leap out of bed, sometimes injuring themselves or their partners. It was first formally described by Carlos Schenk and Mark Mahawald at the University of Minnesota in 1986, and it turned out to be more than just a curiosity. It's now recognized as one of the strongest early predictors of certain neurodegenerative diseases, particularly Parkinson's disease and a related condition called Lewy body dementia. Something like 80% of people diagnosed with REM sleep behavior disorder eventually develop one of these conditions, often years or even decades later, which suggests that the brainstem regions responsible for REM paralysis are among the first to be affected by the neurodegenerative process long before the more obvious symptoms appear. But here's the thing about that first REM period of the night. It's short, maybe 5 or 10 minutes, and then you cycle back down into non-REM sleep again, and the whole sequence repeats, but not identically. As the night progresses, the architecture shifts. Each successive cycle contains less deep, slow-wave sleep and more REM sleep. So the first half of your night is dominated by deep sleep. That's when you're getting most of your physical restoration, your growth hormone release, your immune support. And the second half is dominated by REM sleep, which is when most of your vivid dreaming occurs, and which seems to be particularly important for emotional processing and certain types of memory consolidation. This is why the timing of your sleep matters, not just the duration. If you go to bed at 3 in the morning and sleep until 11, you get 8 hours, sure. But you've missed the early night window when your circadian system is primed for the deepest slow-wave sleep. And if you cut your sleep short by waking up at 5 instead of 7, you're disproportionately losing REM sleep, because that's what dominates the final cycles. Either way, you're not getting the full architecture. You're getting a lopsided version. And all of this, the cycling, the shifting proportions, the paralysis, the temperature drops, the hormone surges, all of it is happening automatically without any conscious effort on your part. Your brain is running an extraordinarily complex program, and you're not even aware of it. Which raises a question that kept sleep researchers busy for decades after Oserinsky and Kleitman's discovery. If sleep is this elaborate, this precisely structured, this metabolically costly, what is it actually for? What is the brain doing during all those hours that's so important it can't be done while you're awake? And the first really compelling answers to that question started emerging from an unexpected direction, from researchers who weren't studying sleep at all, but were studying something that seemed completely unrelated: memory. How the brain forms memories, stores them, and decides which ones to keep. That's where the trail leads. And it's one of the most satisfying detective stories in all of neuroscience. So let's talk about a man named Brenda Milner. Actually, let's talk about her most famous patient first, because his story changed everything. You've probably heard of him, even if you don't realize it. For decades, he was known only by his initials, HM, to protect his privacy. His full name, Henry Molaison, wasn't revealed until after his death in 2008. But during his lifetime, he became the single most studied individual in the history of neuroscience. And what happened to him was a tragedy that accidentally illuminated how memory works, which in turn cracked open one of the deepest mysteries about why we sleep. Henry Molaison was born in 1926 in Hartford, Connecticut. As a child, he began experiencing seizures, minor ones at first, but by his late teens, they had become severe, grand mal seizures that were completely debilitating. He couldn't hold a job. He couldn't live a normal life. The seizures were traced to his medial temporal lobes, and by 1953, when Henry was 27, a neurosurgeon named William Beecher Scoville proposed a radical solution. He would surgically remove the structures in both temporal lobes that seemed to be generating the seizures. This included most of the hippocampus on both sides, along with surrounding tissue. The surgery worked, in a sense. Henry's seizures were dramatically reduced, but something else happened that no one had anticipated. Henry could no longer form new long-term memories. He could remember his childhood. He could recall events from before the surgery. But from August 1953 onward, almost nothing new stuck. He would meet someone, have a perfectly pleasant conversation, and then 5 minutes later, have no memory of ever having met them. He read the same magazines over and over without recognizing them. He could not tell you what year it was, or what he'd had for breakfast, or that his father had died. Every day was a kind of perpetual present tense. This was devastating for Henry, obviously, but for science, it was a revelation, because it proved something that had only been theorized before, that the hippocampus is essential for converting short-term experiences into lasting long-term memories. Without it, you can hold things in your mind for a few seconds, maybe a few minutes, but they never get written into permanent storage. The hippocampus isn't where long-term memories live. They end up distributed across the cortex, but the hippocampus is the gateway. It's the librarian who decides what gets filed and where. Now here's where sleep enters the picture, and this is where it gets really interesting. Brenda Milner, a neuropsychologist at the Montreal Neurological Institute, spent years studying Henry and other patients with similar injuries. And one of the things she and her colleagues noticed was that even in healthy people, memories seemed fragile right after they were formed. You could learn something new, and if you were tested immediately, you'd do okay. But the memory was wobbly. It could be disrupted by interference, by learning something else right afterward, by distraction, by stress. It took time for a new memory to become stable, to become resistant to disruption. This process was called consolidation, and researchers started asking a natural question. When does consolidation happen? What is the brain doing during the hours and days after you learn something that transforms a fragile, temporary trace into a durable, long-term memory? The first hints came from studies in the 1920s, believe it or not. Two psychologists, John Jenkins and Carl Dallenbach, published a beautifully simple experiment in 1924. They had people memorize lists of nonsense syllables, random combinations of letters that don't mean anything, so you can't rely on existing knowledge to remember them. Then they tested recall after different intervals. Here's what they found. People who slept during the interval between learning and testing remembered significantly more than people who stayed awake for the same amount of time. And this wasn't just because being awake meant more interference from new experiences, though that was part of it. There was something about sleep itself that seemed to actively protect or strengthen the memories. But this was the 1920s. Nobody had EEGs yet. Nobody knew about sleep stages. Nobody had the tools to figure out what was actually happening in the brain during sleep. So the finding just kind of sat there, a curious observation without a mechanism. It took decades before anyone could really follow up on it properly. Fast forward to the 1990s and early 2000s. By now, researchers had sophisticated tools, not just EEGs, but functional MRI, which lets you watch blood flow changes in the brain in real time, and techniques for recording from individual neurons in animal brains. And a series of extraordinary experiments started to reveal what the sleeping brain is actually doing with your memories. One of the most striking came from a lab at MIT, from a neuroscientist named Matthew Wilson. In 1994, Wilson and his colleague Bruce McNaughton published a study that honestly still gives me chills when I think about it. They implanted tiny electrodes into the hippocampi of rats, the same structure that Henry Molaison had lost, and recorded the activity of individual neurons while the rats ran through mazes during the day. Specific cells, called place cells, fired in specific patterns depending on where the rat was in the maze. So, as the rat ran a particular route, you'd see a particular sequence of neurons firing, like notes in a melody. Each route had its own signature tune. Then Wilson and McNaughton recorded from the same neurons while the rats slept. And here's the thing that changed everything. During slow-wave sleep, that deep, delta-wave-dominated sleep that dominates the first half of the night, the same stage we talked about earlier, the rats' hippocampal neurons replayed the same firing sequences they had produced while running the maze. The same melody played again. Not randomly, not approximately. The same specific sequences in the same order, just compressed in time. The sleeping brain was literally rehearsing the day's experiences. Think about what that means. While the rat is lying there, eyes closed, body still, its hippocampus is running through the maze again, and again, and again. It's practicing. It's replaying. And this replay isn't just the hippocampus talking to itself. Subsequent studies showed that the hippocampal replay was coordinated with activity in the cortex, the outer layer of the brain where long-term memories are ultimately stored. It was as if the hippocampus was teaching the cortex, playing back the day's recordings, and slowly transferring the information to a more permanent archive. Remember those sleep spindles we talked about? Those brief bursts of activity in stage 2 sleep, generated by the thalamus? It turns out they're not just random neural chatter. Sleep spindles appear to be tightly linked to memory consolidation. They tend to increase after learning, and their timing is coordinated with the slow oscillations of deep sleep and the hippocampal replay events. Some researchers describe it as a kind of 3-part dialogue. The slow cortical oscillation sets the rhythm. The sleep spindle opens a window of plasticity in the cortex, and the hippocampal replay fires the memory content through that window. It's like the brain has evolved an entire communication protocol specifically for transferring memories during sleep. And this isn't just a rat thing. In humans, Jan Born and his colleagues in Germany, Born is one of the giants of sleep and memory research, showed in a series of elegant experiments starting in the early 2000s that human declarative memory, the kind of memory you use for facts and events, is significantly enhanced by sleep, and specifically by slow-wave sleep. They had people learn word pairs before sleep, then tested them afterward. Not only did sleep improve recall compared to an equivalent period of wakefulness, but the amount of improvement correlated with the amount of slow-wave sleep the person got. More deep sleep, better memory. And when they used a clever technique called transcranial direct current stimulation to gently enhance slow oscillations during sleep, memory performance improved even further. They were literally boosting the brain's own consolidation machinery. But here's what's really elegant about this system, and what makes it more than just a simple recording and playback mechanism. The brain doesn't just faithfully copy everything from the day into long-term storage. It's selective. It prioritizes. Memories that are emotionally significant, or that you've been told will be important later, or that connect to things you already know, those get preferentially consolidated during sleep. The brain is editing, it's curating, it's deciding what matters and what can be safely forgotten. And forgetting, it turns out, might be just as important as remembering. There's a theory, championed by Giulio Tononi and Chiara Cirelli at the University of Wisconsin, called the synaptic homeostasis hypothesis. The idea is beautifully simple. During the day, as you learn and experience things, synapses throughout your brain get strengthened. Every new experience, every new piece of information, every new skill, they all involve strengthening connections between neurons. But you can't just keep strengthening synapses indefinitely. The system would saturate. It would become noisy, energy hungry, and eventually unable to learn anything new. So during sleep, particularly during deep slow-wave sleep, the brain globally scales down synaptic strength. It weakens all the connections a little bit. The ones that were strongly reinforced during the day survive this pruning because they're strong enough to withstand the downscaling. But the weak ones, the trivial ones, the noise, those get washed away. It's like writing on a beach. During the day, you're scratching messages in the sand. Some are deep, carved with intention. Others are faint, barely visible. Then sleep comes like a gentle wave, washing over the whole beach. The faint marks disappear. The deep ones remain. And in the morning, what's left is a cleaner, clearer record of what actually mattered. This is why, if you've ever struggled with a problem before bed and then woken up with the answer, it's not just folklore. Your brain was working on it, not consciously, not in a way you could direct or control, but through this process of replay, consolidation, and pruning. The unnecessary details got stripped away, the important connections got strengthened, and sometimes, the solution just emerges. Now, all of this explains a lot about what deep slow-wave sleep is for. But what about REM sleep? Remember, that's the stage where your brain is wildly active, your eyes are darting around, you're dreaming vivid, narrative, emotionally charged dreams, and your body is paralyzed. What's that doing for memory? This is where the story gets more nuanced and honestly more contested. Because while slow-wave sleep seems to be primarily about declarative memory, facts, events, things you can consciously recall, REM sleep appears to be involved in a different kind of memory processing. Procedural memory, for one, the kind of memory you use for skills, like playing piano or riding a bicycle. Several studies have shown that REM sleep deprivation impairs the consolidation of newly learned motor skills, even when total sleep time is kept roughly the same. But the role of REM that's gotten the most attention in recent years is emotional memory processing. And this is where we meet someone whose work has reshaped how we think about the relationship between sleep and mental health. A neuroscientist named Matthew Walker, who you might know from his best-selling book, though his research contributions go much deeper than any popular book could capture. Walker, working at UC Berkeley, proposed something that sounds almost poetic, but is grounded in data, that REM sleep serves as a kind of overnight therapy. During REM, the brain reprocesses emotional memories, but it does so in a neurochemical environment that's fundamentally different from waking life. Specifically, during REM sleep, norepinephrine, which you might know as noradrenaline, the brain's version of adrenaline, the chemical of stress and anxiety and fight or flight, is almost completely absent. It's one of the only times in your entire 24-hour cycle when your brain is essentially free of this stress chemical. And Walker's argument is that this allows the brain to reprocess difficult emotional memories without the emotional sting. You get to revisit the experience, integrate it, extract its meaning, but strip away some of the raw emotional charge. So you wake up and the memory is still there, but it doesn't hurt quite as much. The content is preserved, but the visceral gut punch quality has been softened. And this has profound implications for understanding what goes wrong when sleep is disrupted. Think about post-traumatic stress disorder, for instance. One of the hallmarks of PTSD is that traumatic memories don't fade the way they should. They remain vivid, intrusive, and emotionally overwhelming, sometimes for years. And people with PTSD frequently have disrupted REM sleep. The question researchers are now asking is whether the sleep disruption isn't just a symptom of PTSD, but part of the mechanism, whether the inability to get proper REM sleep prevents the brain from doing its normal emotional processing, which keeps the traumatic memory locked in its raw, unprocessed state. It's a chicken and egg problem that's still being untangled, but the implications are enormous. And this connects to something even broader, something that researchers have only begun to appreciate in the last decade or so: the relationship between sleep and the physical health of the brain itself. Because memory consolidation and emotional processing aren't the only things happening while you sleep. There's a whole other system at work, one that was only discovered in 2012, and when you hear about it, you'll understand why some scientists have called it one of the most important discoveries in neuroscience in the last 50 years. It's called the glymphatic system, and it was found by a Danish neuroscientist working at the University of Rochester named Maiken Nedergaard. And the story of how she found it is one of those beautiful examples of science happening, because someone was willing to look at a familiar problem from a completely unfamiliar angle. Nedergaard had spent years studying glial cells. Those are the non-neuronal cells in your brain, the ones that for a long time were basically dismissed as packing material, the stuff that holds the important cells in place. The word glia literally comes from the Greek word for glue. That's how little respect these cells got. Neurons were the stars, the ones doing all the signaling, all the thinking, all the computing. Glial cells were just there, structural support, background players. But Nedergaard had a hunch that glial cells were doing something far more interesting than anyone gave them credit for, and she'd been chipping away at that idea for years. And in 2012, she and her team published a paper that genuinely changed how we think about what the brain does while you sleep. Here's the problem she was trying to solve. Every organ in your body produces metabolic waste. That's just a fact of being alive. Cells burn energy. They do work. And in the process, they generate byproducts that need to be cleared away. Your body handles this with the lymphatic system, a network of vessels that runs alongside your blood vessels and drains waste products out of tissues and into the bloodstream so they can be filtered and eliminated. It's like the sewage system of your body. Every organ has lymphatic drainage. Every organ except one: the brain. For decades, this was a genuine puzzle. The brain is the most metabolically active organ you have. It's only about 2% of your body weight, but it consumes roughly 20% of your energy. It's producing waste constantly, and yet it has no lymphatic vessels. So how does it take out the trash? The answer, it turned out, was hiding in the cerebrospinal fluid, that clear liquid that bathes your brain and spinal cord. Scientists had known about cerebrospinal fluid for centuries. They knew it cushioned the brain, provided buoyancy, delivered some nutrients. But Nedergaard's team discovered something nobody had seen before. They found that cerebrospinal fluid doesn't just sit passively around the brain. It actually flows through the brain tissue itself, along channels that run parallel to blood vessels, pumped by the pulsing of the arteries. And as it flows through, it picks up waste products, metabolic junk, misfolded proteins, cellular debris, and carries them out. The channels are formed by a particular type of glial cell called astrocytes, which wrap their little feet around blood vessels and create a kind of plumbing network. Nedergaard called it the glymphatic system, a mashup of glial and lymphatic, because it's essentially the brain's own private lymphatic system, built out of glial cells. Now here's where sleep enters the picture, and this is the part that really made people sit up and pay attention. Nedergaard's team was working with mice, and they were using a technique called two-photon microscopy to watch the flow of cerebrospinal fluid through the brain in real time. And they noticed something remarkable. When the mice were awake, the flow was minimal. The channels were narrow, the fluid barely moved. But when the mice fell asleep, the channels opened up dramatically. The interstitial space between brain cells expanded by roughly 60%, and cerebrospinal fluid came flooding through like someone had opened a fire hydrant. The glymphatic system, it turned out, is almost entirely a sleep-time operation. It runs at a trickle when you're awake, and it opens up full blast when you're asleep. Think about what that means. Your brain is essentially running a massive cleaning cycle every time you sleep. All day long, while you're thinking and working and worrying and scrolling through your phone, your brain is accumulating metabolic waste, and it's mostly just sitting there, piling up. And then you fall asleep, and the cleanup crew comes in. The cells actually shrink a little, creating more space between them, and the cerebrospinal fluid rushes through and flushes everything out. It's like a dishwasher that only runs at night. And here's the kicker. One of the key waste products that the glymphatic system clears is beta amyloid. That name might ring a bell. Beta amyloid is a protein that, when it accumulates in the brain, forms the plaques that are one of the hallmark features of Alzheimer's disease. Nedergaard's team showed that beta amyloid is cleared from the brain twice as fast during sleep as during wakefulness. Twice as fast. And this immediately raised a question that researchers are still actively investigating. Could chronic sleep deprivation contribute to the development of Alzheimer's? If you're not sleeping enough and your glymphatic system isn't getting enough time to clear beta amyloid, does that protein build up over time and eventually reach toxic levels? The epidemiological evidence is suggestive, and it's growing. Multiple studies have found that people who consistently sleep less than 6 hours a night have a significantly higher risk of developing Alzheimer's later in life. There's also evidence that sleep quality matters. It's not just about hours in bed. It's about getting enough deep, slow-wave sleep, which is when the glymphatic system appears to be most active. And here's a cruel irony. Alzheimer's disease itself disrupts sleep, particularly slow-wave sleep, which means the disease may accelerate its own progression by impairing the very cleaning mechanism that could slow it down. It's a vicious cycle, and understanding it is one of the most urgent questions in neuroscience right now. Now, I want to be careful here, because the relationship between sleep and Alzheimer's is still being worked out. Correlation isn't causation, and the brain is complicated. But the glymphatic discovery gave researchers a plausible biological mechanism connecting sleep loss to neurodegeneration, and that was a huge deal. Before Nedergaard's work, the link between poor sleep and cognitive decline was mostly observational. People noticed the pattern, but nobody could explain why it would be the case. The glymphatic system provided a why, and it added yet another layer to the growing case that sleep isn't a luxury, isn't downtime, isn't the brain idling. It's an active, essential biological process, and skipping it has real, measurable, physical consequences. So let's step back for a moment and appreciate where we are. We've talked about sleep consolidating your memories, pruning your synapses, processing your emotions, and now physically cleaning your brain. That's an extraordinary amount of work for something that looks, from the outside, like you're just lying there, doing nothing. And all of these processes are happening in an orchestrated sequence across the night, tied to specific sleep stages regulated by the interplay of your circadian clock and your homeostatic sleep drive. It's an incredibly sophisticated system, which makes it all the more remarkable how badly we can mess it up. And that brings us to something that affects almost every person listening to this right now: the modern world's assault on sleep. Because here's the thing. The biological machinery of sleep evolved over hundreds of millions of years in environments that looked nothing like the one you live in today. Your circadian system was calibrated by the sun. Bright light during the day, darkness at night. Your ancestors didn't have electric lights, didn't have screens, didn't have shift work, didn't have transatlantic flights. And when you think about it that way, it's honestly amazing that our sleep systems work as well as they do in the modern world, because almost everything about contemporary life is designed, inadvertently, to interfere with them. Let's start with light, because that's the big one. Remember the melanopsin-containing retinal ganglion cells we talked about earlier? Those specialized cells in your eyes that detect ambient light levels and send signals to your suprachiasmatic nucleus to set your circadian clock. Those cells are particularly sensitive to short wavelength light, blue light, in the range of about 460 to 480 nanometers. And guess what kind of light your phone screen emits? Your tablet, your laptop, your LED television. Blue light, lots of it, right into your eyes, often right before bed. In 2014, a team led by Anne-Marie Chang at Harvard published a study in the Proceedings of the National Academy of Sciences that compared people reading on an iPad before bed to people reading a printed book. The iPad readers showed suppressed melatonin secretion. Their melatonin release was delayed by about an hour and a half. They took longer to fall asleep. They had less REM sleep. And this is the part that really matters for daily life, they were sleepier and less alert the next morning, even after getting the same amount of time in bed. The light from the screen was literally shifting their circadian clock later, telling their brain it wasn't nighttime yet, when it absolutely was. And this isn't just about screens. The broader issue is that we live in a world saturated with artificial light. Before Edison, the average person experienced about 10 to 13 hours of darkness every night, depending on the season. Now, most people in industrialized countries get maybe 6 or 7 hours of true darkness, if that. And even that is often interrupted. Streetlights through the curtains, the standby glow of electronics, a bathroom light at 3 in the morning. Your suprachiasmatic nucleus is getting mixed signals all night long. But light is only part of the story. There's also caffeine, which we touched on earlier, adenosine's antagonist, the molecule that blocks your sleepiness signals without actually clearing the adenosine from your system. The half-life of caffeine in the human body is roughly 5 to 6 hours, which means that if you have a cup of coffee at 4 in the afternoon, by 10 at night, you still have about half of that caffeine circulating in your bloodstream, sitting on your adenosine receptors, whispering to your brain that you're not as tired as you actually are. And plenty of people have caffeine much later than 4. Some people have it after dinner. Some people have it without even realizing it. Chocolate, certain teas, some pain medications, even decaf coffee contains small amounts. Then there's alcohol, and this one is sneaky because alcohol feels like it helps you sleep. And in a narrow sense, it does. Alcohol is a sedative, and it will help you fall asleep faster. But what it does to your sleep architecture is devastating. Alcohol suppresses REM sleep, sometimes profoundly. It fragments your sleep in the second half of the night as your body metabolizes it. It relaxes the muscles of your airway, making snoring and sleep apnea worse. And it blocks the normal pattern of sleep cycles, so even if you spend 8 hours in bed after a few drinks, The sleep you get is qualitatively different, shallower, more disrupted, less restorative. You wake up feeling unrested, and there's a real physiological reason for that. The sleep you got wasn't doing all the things sleep is supposed to do. And then there's something that doesn't get talked about enough, which is temperature. Your body needs to drop its core temperature by about 1 to 1.5 degrees Celsius to initiate and maintain good sleep. This is part of your circadian rhythm. Your body temperature naturally peaks in the late afternoon and drops to its lowest point in the early morning hours. And this temperature drop isn't just correlated with sleep, it actually helps trigger it. Your body dumps heat through your hands and feet. That's why your extremities often feel warm when you're getting drowsy, because blood is flowing to the surface to radiate heat away. But if your bedroom is too warm, if you're under too many blankets, if your thermostat is set to 72 degrees, your body can't shed that heat effectively, and your sleep suffers. Most sleep researchers recommend a bedroom temperature of around 65 degrees Fahrenheit, about 18 Celsius, which sounds cold to a lot of people, but your biology is pretty clear on this point. So you've got light disruption, caffeine interference, alcohol damage, temperature problems, and we haven't even gotten to the psychological side yet, because there's another modern phenomenon that's become one of the most common sleep disorders in the world, and it's fundamentally a disorder of the mind trying too hard to do something that should be automatic. It's called insomnia, and the story of how we came to understand it, and more importantly, how we learned to treat it without drugs, starts with a psychologist named Arthur Spielman, who in 1987 proposed something he called the three-factor model. And it's one of those frameworks that's so elegant and so useful that once you hear it, you'll never think about sleeplessness the same way again. Spielman argued that insomnia isn't one thing. It's the result of 3 separate forces that stack on top of each other. He called them predisposing factors, precipitating factors, and perpetuating factors. And the beauty of this model is that it explains why insomnia starts, why it sticks around, and crucially, why it often persists long after the original cause is gone. So let's walk through it. Predisposing factors are the things about you that make you more vulnerable to insomnia in the first place. Maybe you're someone who's naturally a light sleeper. Maybe you have a tendency toward anxiety or rumination. Your mind just likes to chew on things. Maybe you have a genetic predisposition toward hyperarousal, meaning your nervous system runs a little hotter than average, a little more vigilant. These factors alone don't necessarily cause insomnia. Plenty of people with anxious temperaments sleep fine most of the time, but they lower the threshold. They make you more susceptible. Then come the precipitating factors, the triggers. Something happens in your life that disrupts your sleep. A stressful job change, a divorce, a health scare, the death of someone close to you, a new baby, even something positive, like a big promotion that comes with more pressure. These events cause acute insomnia. A few bad nights, maybe a few bad weeks. And here's the thing. For most people, once the stressor resolves or they adapt to it, their sleep goes back to normal. The precipitating factor fades and the system resets. But for some people, especially those with higher predisposing vulnerability, something else kicks in. And this is where Spielman's real insight lives. The perpetuating factors. These are the behaviors and thought patterns that people develop in response to their sleep problems. Behaviors that seem helpful in the moment, but actually make the insomnia worse and keep it alive long after the original trigger is gone. And this is so common, it's almost universal among chronic insomnia sufferers. Here's what happens. You have a few bad nights. You start worrying about sleep. You start going to bed earlier, hoping to catch up. You start lying in bed for 9 or 10 hours even though you're only sleeping 5 or 6. You start napping during the day. You start watching the clock at 3 in the morning, calculating how many hours you have left. You start associating your bed, the physical space, the pillow, the sheets, with frustration and anxiety instead of rest. And every one of these responses, which feel completely logical when you're exhausted and desperate, actually trains your brain to be awake in bed. You're conditioning yourself, in the most Pavlovian sense, to associate your sleep environment with wakefulness and stress. Think about what that means. Your bed becomes a cue for arousal instead of a cue for sleep. And the harder you try to sleep, the more elusive it becomes. Because trying to sleep is an act of effort, and effort is the opposite of the letting go that sleep requires. It's like trying to force yourself to relax. The very act of trying creates tension. Spielman saw this pattern over and over, and his 3-factor model gave clinicians a way to understand why someone whose original stressor was a difficult work project 6 months ago is still lying awake at 2 in the morning long after that project is finished. The precipitating factor is gone, but the perpetuating factors have taken over. The insomnia has become self-sustaining. And this understanding, that chronic insomnia is maintained by learned behaviors and conditioned associations, opened the door to what many sleep researchers now consider the most important therapeutic development in the history of sleep medicine. It's called cognitive behavioral therapy for insomnia, or CBTI, and it was developed and refined through the late 1980s and 1990s by researchers including Spielman himself, along with Charles Morin at Université Laval in Quebec and Jack Ettinger at Duke University, among others. And what makes CBTI so remarkable is that it works better than sleeping pills. Not just as well, better. And it works longer. And it does it without any of the side effects. Here's how it works, broadly. CBTI has several components, and they attack the perpetuating factors from different angles. The first, and probably the most counterintuitive, is something called sleep restriction therapy. And this is the one that makes people's eyes go wide when they first hear about it. Because if you're someone who's struggling to sleep, the last thing you want to hear is that your therapist is going to reduce the amount of time you're allowed to spend in bed. But that's exactly what happens. The logic is beautifully simple. If you're spending 9 hours in bed but only sleeping 5, your sleep efficiency, the ratio of time asleep to time in bed, is terrible, around 55%. So the therapist restricts your time in bed to match the amount you're actually sleeping, maybe 5.5 hours. You pick a fixed wake time, say 6 in the morning, and you're not allowed to get into bed until 12:30. That's it. That's your sleep window. Now, the first few nights are rough. You're sleep deprived and you're not going to bed until after midnight. But here's what happens. Because you're building up so much sleep pressure, remember all that adenosine accumulating? When you finally do get into bed, you fall asleep fast, and you sleep more solidly. Your sleep efficiency shoots up. Your brain starts relearning that bed equals sleep, not bed equals lying awake staring at the ceiling. Once your efficiency gets above 85 or 90%, the therapist gradually expands the sleep window. 15 minutes earlier to bed, then another 15 minutes. Slowly, methodically, you rebuild a solid, consolidated sleep pattern. You're reconditioning your brain. The second component is stimulus control, which was actually developed even earlier, in the 1970s, by a psychologist named Richard Bootson at the University of Arizona. Stimulus control is all about breaking those negative associations with the bed. The rules are simple but strict. You only go to bed when you're sleepy. If you're in bed and you're not asleep within roughly 15 or 20 minutes, you get up and go to another room and do something quiet and boring until you feel sleepy again. You don't read in bed. You don't watch TV in bed. You don't scroll your phone in bed. The bed is for sleep and intimacy, and that's it. And you keep a fixed wake time regardless of how poorly you slept. No sleeping in, no naps. You're systematically retraining your brain to associate the bed with rapid sleep onset. Then there's the cognitive piece, the C in CBT. This targets the anxious, catastrophic thinking that insomnia breeds. Thoughts like, if I don't sleep tonight, I won't be able to function tomorrow. Or, I haven't slept well in weeks, my health must be deteriorating. Or, I'm one of those people who just can't sleep. A CBTI therapist helps you examine these thoughts, test them against reality, and replace them with more accurate, less panic-inducing beliefs. Because the truth is, most people with insomnia actually sleep more than they think they do. There's a well-documented phenomenon called sleep state misperception, where people in a sleep lab who report lying awake all night turn out to have slept for 5 or 6 hours. Their perception of wakefulness is distorted by the anxiety and hyperarousal. And while chronic short sleep is genuinely bad for you, one bad night, or even several, is not the catastrophe your anxious brain tells you it is. Humans are remarkably resilient to short-term sleep loss. Your performance dips, sure, but you don't collapse. Now here's what really convinced the medical establishment. In 1999, Charles Moran published a landmark study comparing CBTI to pharmacotherapy, specifically the benzodiazepine receptor agonist temazepam, in older adults with chronic insomnia. Both treatments worked in the short term. But when Moran followed up months later, the CBTI group had maintained their improvements, while the medication group had relapsed. The drugs worked while you took them, but they didn't teach your brain anything. CBTI actually changed the underlying patterns, and this finding has been replicated over and over. A major meta-analysis published in 2015 in the Annals of Internal Medicine, commissioned by the American College of Physicians, reviewed the entire evidence base and concluded that CBTI should be the first-line treatment for chronic insomnia in adults, not medication, not supplements, not anything else. CBTI first. And yet, and this is one of those frustrating gaps between what science knows and what medicine does, most people with insomnia still get prescribed a pill. Part of the problem is access. There simply aren't enough trained CBTI therapists. As of the early 2020s, there were only a few thousand board-certified behavioral sleep medicine specialists in the United States, serving a population where roughly 10% of adults meet criteria for chronic insomnia disorder. That's a staggering mismatch, which is why there's been a big push to develop digital CBTI programs, apps, and online platforms that deliver the therapy through automated modules. One of the most studied is called Sleepio, developed by Colin Espy at the University of Oxford, and randomized trials have shown it produces clinically meaningful improvements comparable to face-to-face therapy. It's not perfect, and some people need the human connection of a real therapist, but it's dramatically expanding access. So that's insomnia, a disorder that turned out to be far more about learned behavior and conditioned arousal than about any fundamental biological defect, and one that responds beautifully to a treatment that essentially re-educates the sleeping brain. But insomnia is only one piece of the modern sleep disorder landscape, and there are other conditions that are far more biologically rooted, far harder to treat, and in some cases, genuinely dangerous. And one of the most fascinating, and for a long time, one of the most mysterious, is narcolepsy. Because narcolepsy isn't just about being excessively sleepy during the day, although that's certainly part of it. It's about a fundamental breakdown in the brain's ability to regulate the boundaries between sleep states, and the story of how researchers finally cracked its mechanism. In the late 1990s, involves two independent labs racing toward the same molecule from completely different directions, and neither team knew the other one existed until the papers landed almost simultaneously. So let's talk about that. The first lab was led by Luis de la Sea and Thomas Kilduff, working at the Scripps Research Institute in San Diego. They were doing something fairly routine in neuroscience, hunting for new neuropeptides, which are small signaling molecules that neurons use to communicate with each other. In 1998, they identified a pair of peptides produced by a tiny cluster of neurons in the lateral hypothalamus, and they named them hypocretins because they were found in the hypothalamus, and they bore a faint structural resemblance to the gut hormone secretin. They published their discovery, noted that these neurons seemed to be involved in arousal and feeding behavior, and moved on to characterize them further. Meanwhile, at the University of Texas Southwestern Medical Center in Dallas, a researcher named Masashi Yanagisawa and his team were independently hunting for ligands, molecules that bind to orphan receptors, which are receptors in the brain whose natural chemical partners hadn't been identified yet. They found the exact same pair of peptides, but they gave them a completely different name. Yanagisawa called them orexins, from the Greek word orexis, meaning appetite, because their early experiments suggested a role in feeding and energy regulation. Orexin A and orexin B. Same molecules, different names. And to this day, both names are used in the literature, which can be confusing, but that's science for you. Sometimes two groups find the same thing at the same time, and the naming never quite gets sorted out. Now here's where it gets really interesting. In 1999, just a year after both teams published their discovery papers, Yanagisawa's group created a line of knockout mice genetically engineered to lack the gene for orexin. And these mice did something extraordinary. They would be walking around, eating, behaving normally, and then suddenly collapse. They'd just drop. Their muscles would go limp, their eyes would close, and within seconds, they'd enter REM sleep. Then, just as abruptly, they'd wake up and go about their business as if nothing had happened. If you've ever seen a video of this, it's genuinely startling. The mice looked exactly like narcoleptic humans experiencing cataplexy, that sudden loss of muscle tone triggered by emotion that's one of the hallmark symptoms of narcolepsy. And almost simultaneously, Emmanuel Mignot at Stanford, who had spent over a decade studying a colony of narcoleptic Doberman pinschers. Yes, dogs can have narcolepsy, and these Dobermans were famous in sleep research. Mignot finally identified the genetic mutation responsible for their condition. It was a mutation in the orexin receptor 2 gene. The dog's brains couldn't properly receive the orexin signal. Different angle, same system, same conclusion. Narcolepsy, at least the most common and severe form now called type 1 narcolepsy, was a disease of orexin deficiency. Think about what that means for a second. For decades, narcolepsy had been this baffling clinical puzzle. People knew it ran in families somewhat. They knew it involved excessive daytime sleepiness and these bizarre intrusions of REM sleep into waking life, but nobody understood the mechanism. And then, within the span of about 18 months, 3 independent lines of evidence, de la Sea and Kilduff's peptide discovery, Yanagisawa's knockout mice, and Mignot's narcoleptic dogs, all converged on the same tiny population of neurons in the lateral hypothalamus producing the same molecule. It was one of those beautiful moments in science where everything clicks. And here's the thing that made it even more poignant. When researchers examined the brains of humans who had died with narcolepsy, they found that those orexin-producing neurons were almost completely destroyed. We're talking about a population of only roughly 70,000 neurons in the human brain, a minuscule number out of the 86 billion or so total. And in narcoleptic patients, 85 to 95% of them were gone. The leading hypothesis, which has gained substantial support over the years, is that narcolepsy type 1 is an autoimmune disease. The body's own immune system, for reasons still not entirely understood, selectively destroys those orexin neurons. There's a strong genetic association with a specific immune system gene variant called HLA-DQB1 06.02, which is present in over 98% of people with type 1 narcolepsy. That's one of the tightest HLA associations with any disease in medicine. And there have been epidemiological clues, a spike in narcolepsy cases among children in Scandinavia following a particular H1N1 flu vaccine called Pandemrix in 2009 and 2010 suggested that in genetically susceptible individuals, an immune trigger could set off the autoimmune attack on those orexin cells. It was a tragedy for those affected families, but it provided a crucial piece of the puzzle. So narcolepsy became, in a sense, a story about a single molecule and a tiny cluster of neurons that punch far above their weight. Those 70,000 orexin neurons act as a kind of stabilizer for the sleep-wake switch. Remember how we talked about the flip-flop model? The idea that the brain doesn't gradually slide between sleep and wakefulness, but rather flips between them like a light switch. The orexin neurons are what keep that switch locked in the wake position during the day. They project widely throughout the brain, sending excitatory signals to all the major arousal centers, the locus coeruleus, the dorsal raphe, the tuberomammillary nucleus, the basal forebrain. Without orexin, the switch becomes unstable. It can flip to sleep at inappropriate times, and worse, it can flip into REM sleep directly, which is what produces the most dramatic symptoms of narcolepsy. Cataplexy, for instance, that sudden muscle weakness triggered by strong emotions like laughter or surprise, is essentially REM atonia intruding into wakefulness. Remember the sublaterodorsal nucleus and how it paralyzes your voluntary muscles during REM sleep to keep you from acting out your dreams. In narcolepsy, that paralysis mechanism can activate while you're still conscious. You're standing there laughing at a joke and your knees buckle because your brainstem just triggered the REM atonia circuit. Sleep paralysis, another common narcolepsy symptom, is the same mechanism firing at the wrong time. You wake up, but the atonia hasn't released yet, so you're conscious but temporarily unable to move. And hypnagogic hallucinations, those vivid dreamlike experiences at sleep onset, are REM dream imagery bleeding into the transition from wake to sleep. All of these symptoms make perfect sense once you understand that narcolepsy is fundamentally a disorder of state boundary regulation, and that orexin is the molecule that normally keeps those boundaries clean and sharp. Now, this understanding led directly to a new class of medications, and this is one of those cases where basic science translated into clinical treatment in a surprisingly elegant way. If narcolepsy is caused by a loss of orexin, the obvious therapeutic goal is to replace it. And while orexin replacement therapy has been challenging, the peptides don't cross the blood-brain barrier easily. Researchers have been developing orexin receptor agonists, small molecules that can mimic orexin's effects. But here's what's kind of ironic and wonderful. The pharmaceutical path that actually reached patients first went in the opposite direction. Drug companies realized that if blocking orexin could destabilize the wake state, then an orexin receptor antagonist, a drug that blocks orexin signaling, could be used as a sleeping pill. And that's exactly what happened. In 2014, the FDA approved suvorexant, sold as Belsomra, the first orexin receptor antagonist for the treatment of insomnia. A few years later came lemborexant. These drugs work by temporarily blocking the orexin system, essentially telling those arousal-promoting circuits to quiet down, which allows the sleep drive to take over more naturally than older sedatives like benzodiazepines, which just broadly dampen brain activity. So the same molecule that explained narcolepsy also gave us a fundamentally new kind of sleeping pill. That's the kind of thing that makes you appreciate how interconnected sleep science really is. One discovery branches in multiple directions. But let's come back to narcolepsy for a moment, because the human side of this story matters. Narcolepsy affects roughly 1 in every 2,000 people, and the average time from symptom onset to correct diagnosis has historically been somewhere between 8 and 15 years. Think about that. A teenager starts falling asleep in class, and everyone assumes they're lazy or staying up too late. They experience cataplexy, and people think they're faking it or having seizures. The condition typically begins in adolescence or early adulthood, right when social and academic pressures are highest, and the stigma and misunderstanding can be devastating. Mignot himself has spoken about patients who went decades without a diagnosis, who lost jobs, relationships, and years of their lives to a condition that was always biological, always neurological, always rooted in the destruction of a specific cell population, but was dismissed as a character flaw. And narcolepsy connects to something broader about how we think about sleep disorders in general, which is that many of them sit at the intersection of neurology, psychiatry, and general medicine in ways that make them easy to miss. Take another condition that's far more common and arguably even more consequential from a public health standpoint: obstructive sleep apnea. If narcolepsy is a story about a missing molecule, sleep apnea is a story about physics. It's about airflow, tissue mechanics, and the simple engineering problem of keeping a collapsible tube open when the muscles that normally hold it rigid go slack during sleep. Your upper airway, the space behind your tongue and soft palate, the pharynx, is essentially a flexible tube made of soft tissue and muscle. When you're awake, the muscles of the pharynx actively hold it open. But during sleep, especially during deeper stages, muscle tone decreases throughout the body, and the pharynx can narrow or collapse entirely. When it narrows enough to cause turbulent airflow, you get snoring. When it closes completely, you get an apnea, a cessation of breathing that can last 10, 20, 30 seconds or more, until the brain detects the dropping oxygen levels and rising carbon dioxide and triggers a brief arousal, just enough to restore muscle tone and reopen the airway. The person gasps, takes a few breaths, falls back asleep, and the cycle repeats. In severe cases, this can happen 60, 70, even 100 times per hour, every hour, all night long. And most people with sleep apnea have no idea it's happening. They don't remember the arousals. They just wake up feeling exhausted, with a headache, with a dry mouth, and they can't figure out why they're so tired all the time. The consequences go far beyond daytime sleepiness, though. Each one of those apnea events is a miniature physiological crisis. Oxygen levels drop. The sympathetic nervous system fires, blood pressure spikes, heart rate surges, stress hormones flood the bloodstream. Night after night, year after year, this takes a cumulative toll on the cardiovascular system that's genuinely frightening. Untreated severe obstructive sleep apnea roughly doubles the risk of hypertension, significantly increases the risk of atrial fibrillation, stroke, heart attack, and type 2 diabetes. There's even emerging evidence linking it to accelerated cognitive decline, which makes sense when you think about the glymphatic system. If your sleep is being fragmented dozens of times per hour, that waste clearance process we talked about earlier never gets the sustained deep sleep it needs to operate efficiently. The gold standard treatment remains continuous positive airway pressure, CPAP, a device that delivers a gentle stream of pressurized air through a mask, essentially acting as a pneumatic splint to keep the airway open. It was first developed by Colin Sullivan, an Australian physician, in 1981, and the early prototypes were literally built from vacuum cleaner motors and industrial tubing. Sullivan was working at the Royal Prince Alfred Hospital in Sydney, and the story goes that he rigged up this contraption in his lab, a reversed vacuum cleaner motor pushing air instead of pulling it, connected to a length of flexible tubing and a fiberglass mask that he essentially glued together and sealed against the patient's face with silicone. It looked absurd. It sounded absurd, but it worked. The very first patient Sullivan tried this on was a man with severe obstructive sleep apnea, someone who'd been stopping breathing hundreds of times a night. And when Sullivan turned on that jury-rigged motor and the pressurized air started flowing, the man's airway stayed open. He slept through the night. His oxygen levels stayed stable. The arousal stopped. And when he woke up the next morning, he reportedly said he felt better than he had in years. Just like that. Now, Sullivan published his results in The Lancet in 1981, and you'd think the medical world would have jumped on this immediately, right? A simple, non-surgical treatment for a condition that was destroying people's cardiovascular health and quality of life. But the reception was lukewarm at best. Part of the problem was that sleep apnea itself was still poorly understood and underdiagnosed. A lot of physicians didn't take it seriously. Snoring was considered a nuisance, not a medical condition, and the idea that someone could stop breathing dozens of times per hour without knowing it seemed implausible to doctors who hadn't spent time in a sleep lab watching it happen. And then there was the device itself. Even as manufacturers started producing commercial CPAP machines in the mid-1980s, the masks were bulky, uncomfortable, and loud. Patients hated them. Compliance was terrible. People would try CPAP for a week, find the mask intolerable, shove the machine in a closet, and never touch it again. This is one of those fascinating cases where the engineering challenge wasn't inventing the solution. Sullivan had already done that, but making the solution tolerable enough that people would actually use it. And that took decades of incremental refinement. Quieter blowers, softer mask materials, nasal pillows instead of full face masks, heated humidifiers to prevent the dry mouth and nasal congestion that drove so many patients crazy, auto-titrating machines, called auto-PAP or APAP, that could sense changes in airflow resistance and adjust the pressure breath by breath instead of delivering a fixed pressure all night. Each of these improvements nudged compliance rates upward, slowly, stubbornly. Even today, though, adherence remains one of the biggest challenges in sleep medicine. Studies consistently show that somewhere between 30 and 50% of patients prescribed CPAP don't use it consistently, which is a staggering number when you consider the cardiovascular consequences of leaving severe apnea untreated. And this brings us to something broader, something that connects to almost everything we've been talking about tonight. Because the story of CPAP compliance is really a story about the gap between what we know about sleep and what we actually do about it. And that gap is enormous. We understand more about sleep science now than at any point in human history. We know what sleep stages look like. We know what drives them. We know what they're for. We know what happens when they're disrupted. We have effective treatments for insomnia, for narcolepsy, for sleep apnea. And yet, as a society, we are sleeping worse than ever. Here's the thing. The average self-reported sleep duration in the United States has been declining for decades. In 1942, a Gallup poll found that Americans reported sleeping an average of about 7 hours and 54 minutes per night. By 2013, that number had dropped to 6 hours and 51 minutes. Now, self-reported data is always a little unreliable. People aren't great at estimating how much they actually sleep. But the trend is consistent across multiple studies and multiple countries. And it's not just duration, it's timing, it's quality, it's regularity. We live in a world that is, in many ways, actively hostile to healthy sleep, and most of the reasons are things we've already touched on. Artificial light, caffeine culture, screen time, shift work, the glorification of busyness, the quiet, pervasive belief that sleep is somehow optional, that it's dead time, wasted time, time that could be better spent being productive. Think about how we talk about sleep in everyday language. I'll sleep when I'm dead. You snooze, you lose. We admire people who claim to get by on 4 or 5 hours a night, CEOs, politicians, surgeons, as if needing less sleep is a sign of strength or ambition rather than, in most cases, either a genetic rarity or a delusion. Because here's what the research actually shows. The percentage of the population that can genuinely function well on less than 6 hours of sleep, without any measurable cognitive or health impairment, is vanishingly small. There's a specific gene variant, a mutation in DEC2, identified by Yinghui Fu at the University of California, San Francisco, in 2009, that allows some people to sleep about 6 hours and wake up naturally refreshed. But this mutation is extraordinarily rare. Fu's team estimated it occurs in fewer than 1% of people who claim to be short sleepers. The rest? They're just sleep deprived and have gotten so used to the feeling that they've forgotten what being fully rested actually feels like. And that's one of the most insidious things about chronic sleep deprivation. You adapt to it subjectively without actually adapting to it biologically. There's a landmark study by Hans van Dongen and David Dinges, published in 2003, that demonstrated this beautifully. They took healthy adults and restricted them to either 4, 6, or 8 hours of sleep per night for 2 weeks while testing their cognitive performance daily. The results were striking. The people sleeping 4 hours a night showed dramatic declines in reaction time, attention, and working memory. After 2 weeks, their performance was as bad as someone who'd been awake for 48 hours straight. The 6-hour group declined too, more slowly, but just as steadily. And by the end of 2 weeks, they were performing as poorly as someone who'd been awake for 24 hours. But here's the kicker. When the researchers asked the sleep-restricted subjects how sleepy they felt, their subjective ratings plateaued after a few days. They stopped feeling sleepier, even as their actual performance kept getting worse. They had no idea how impaired they were. They felt fine. They weren't fine. This has real-world consequences that go way beyond feeling groggy. The National Highway Traffic Safety Administration estimates that drowsy driving causes roughly 100,000 police-reported crashes in the United States every year, resulting in over 1,500 deaths and tens of thousands of injuries. And those numbers are almost certainly underestimates, because unlike alcohol, there's no breathalyzer for sleepiness. There's no blood test. When a driver falls asleep at the wheel and crosses the center line, the accident report often just says driver error or unknown cause. the Chernobyl disaster, the Exxon Valdez oil spill, the Challenger space shuttle decision. All of these have been linked, at least in part, to critical decisions made by people who were severely sleep-deprived. The engineers and operators involved weren't careless or incompetent. They were exhausted. Their brains were running on fumes, and in each case, the consequences were catastrophic. Now, you might think that medicine, of all fields, would take sleep deprivation seriously. And in terms of research, it does. But in terms of practice, medical training has historically been one of the worst offenders. The tradition of the 36-hour shift for resident physicians goes back to William Halsted at Johns Hopkins in the late 1800s, and Halsted, this is darkly ironic, was addicted to cocaine for much of his career, which may explain his superhuman endurance expectations. For over a century, the medical profession essentially said, yes, we know sleep deprivation impairs judgment, but our trainees need to learn to push through it. It wasn't until 2003 that the Accreditation Council for Graduate Medical Education in the United States limited resident work hours to 80 per week, 80 hours, which still allows for shifts of up to 24 hours, plus a 6-hour transition period. And even those limits are controversial and inconsistently enforced. So we have this strange paradox. The science is clear. We understand the mechanisms of sleep at a level of detail that would have been unimaginable to Kleitman or Azarinsky. We know about adenosine accumulation and circadian rhythms and orexin stabilization and glymphatic clearance and memory consolidation and synaptic homeostasis. We know that chronic sleep restriction increases the risk of obesity, diabetes, cardiovascular disease, depression, anxiety, and all-cause mortality. We know that it impairs immune function. There's a striking study by Arik Prather at UCSF showing that people who sleep less than 6 hours a night are 4 times more likely to catch a cold when exposed to a rhinovirus than people who sleep more than 7 hours. We know all of this. And yet the structural incentives of modern life push relentlessly in the opposite direction. Let's talk about what's happening at the frontier, though, because this is where things get genuinely exciting. The same molecular tools that revealed the orexin system and the glymphatic pathway are now opening up entirely new questions about sleep that we couldn't even have formulated 20 years ago. Take the question of local sleep, for instance. We've been talking about sleep as if it's a whole brain phenomenon. You're either asleep or you're awake. But it turns out that's an oversimplification. Research by Giulio Tononi, the same Tononi behind the synaptic homeostasis hypothesis, has shown that individual cortical columns can enter sleep-like states independently. In rats that have been kept awake for extended periods, you can find small patches of cortex that start producing slow waves characteristic of deep sleep even while the animal is still technically awake and moving around. The neurons in those patches essentially go offline for brief moments, and during those micro-sleep episodes in those local patches, the rats make errors. They drop food pellets, they stumble. The rest of the brain is awake, but those little islands of cortex have given up and gone to sleep on their own. This might explain something you've probably experienced yourself. You know that feeling when you've been awake too long and you read the same paragraph 3 times without absorbing it? Or you're driving and you realize you have no memory of the last few miles. Your brain wasn't fully asleep. You were still upright, still functioning at some level, but parts of your cortex may have been flickering in and out of a sleep-like state. It's a deeply unsettling idea when you think about it. The boundary between sleep and wakefulness isn't a clean line. It's fuzzy. It's negotiable. Different parts of your brain can be in different states at the same time. And this connects to another frontier that's been gathering momentum over the last decade or so: the relationship between sleep and the immune system at a level of detail that goes far beyond get enough rest when you're sick. Researchers like Luciana Basania and John Bourne have been mapping out how sleep actively orchestrates immune memory, not just in the metaphorical sense, but in a way that parallels how sleep consolidates declarative memories. During slow-wave sleep, the redistribution of immune cells and the hormonal milieu, low cortisol, high growth hormone, elevated prolactin, create conditions that favor the formation of long-term immunological memories. T cells that encountered a pathogen during the day are more effectively consolidated into memory T cells during sleep. It's as if sleep does for your immune system what it does for your hippocampus. It takes the day's experiences and writes them into long-term storage, which raises an extraordinary question, one that researchers are only beginning to grapple with. What happens to your immune defenses when you don't sleep? Not in the vague, hand-wavy sense of you might catch a cold, but in precise, measurable immunological terms. And the answer, it turns out, is striking. One of the clearest demonstrations came from a study led by Arik Prather at the University of California, San Francisco, published in 2015. Prather and his colleagues did something beautifully straightforward. They recruited over 150 healthy volunteers, tracked their sleep using wrist actigraphy for a week to get a real picture of how much they actually slept, not how much they said they slept, but how much they genuinely slept, and then quarantined them in a hotel and squirted live rhinovirus directly into their noses. Cold virus, right up the nose. And then they waited. They monitored everyone for 5 days, testing for infection, and the results were remarkably clean. People who slept fewer than 6 hours a night were 4.2 times more likely to develop a cold than people who slept more than 7 hours. 4.2 times. That's not a subtle statistical nudge. That's a massive effect size, and it held up after controlling for all the usual confounders: stress, smoking, alcohol, body mass index, season, baseline antibody levels. Sleep duration was the dominant predictor. Your nightly sleep was a better predictor of whether you'd get sick than almost anything else they measured. And this connects back to what Bourne's group has been showing about immunological memory consolidation during slow-wave sleep. It's not just that sleep deprivation makes you feel lousy and run down. It's that sleep is an active phase of immune processing. When you cut it short, you're not just tired, you're immunologically compromised in a specific, mechanistic way. Your body had encounters with pathogens during the day, and it needed that slow-wave sleep window to properly consolidate those encounters into lasting immune memory. Skip the sleep, and the immune system's filing process gets disrupted, just like pulling an all-nighter before an exam disrupts your hippocampal memory consolidation. The parallel is almost eerie. And here's another piece of this puzzle that's worth sitting with. Researchers have also found that sleep deprivation reduces the effectiveness of vaccines. A study by Karine Spiegel and colleagues, published back in 2002, showed that men who were limited to 4 hours of sleep per night for 6 nights before receiving a flu vaccine produced less than half the antibody response compared to men who slept normally. Less than half. So the vaccine went in. The immune system encountered the antigen, but without adequate sleep, the body couldn't mount a full response. The implications of that are enormous when you think about public health on a population scale. Millions of people getting vaccinated while chronically sleep-deprived, potentially getting a diminished benefit. It's one of those findings that makes you rethink something as routine as the timing of a flu shot. So, when we zoom out and look at the whole landscape, the glymphatic clearance system washing away metabolic waste during deep sleep, the synaptic homeostasis that Tononi and Sorelli described, where sleep prunes and refines neural connections, the memory consolidation processes that Bourne and Walker have illuminated, and now this immune orchestration. What emerges is a picture of sleep as perhaps the single most complex and multifunctional biological process we engage in. It's not one thing. It's dozens of things happening simultaneously, each dependent on specific sleep stages, specific timing, specific durations. And every one of those processes suffers when sleep is curtailed. Which brings us, in a way, full circle. Because remember how we started this whole journey? We talked about how, for most of human history, sleep was a mystery. Aristotle thought it had something to do with vapors rising from digestion. For centuries, people assumed sleep was simply the absence of wakefulness, a blank, a void, a little death, as the poets like to say. And then, piece by piece, researcher by researcher, the picture filled in. Berger's squiggly lines on paper in the 1920s. Oserinsky's accidental discovery of REM in the 1950s. Sifra sitting alone in a cave, discovering that the body keeps its own time. Rechtschaffen's rats dying from total sleep deprivation without any single identifiable cause of death, as if everything just fell apart at once. Nedergaard's glymphatic system, revealing that the brain literally cleans itself during sleep. Each discovery peeled back another layer, and what was underneath was always more complex, more beautiful, and more essential than anyone had guessed. And now, in the 2020s, we're in this fascinating moment where the field is converging on a kind of unified understanding. Not a single theory of sleep, that's probably too much to hope for, given how many functions sleep serves, but a framework that connects the pieces. The two-process model that Borbély gave us in 1982 still holds as the scaffolding. Circadian rhythm and sleep pressure, process C and process S, working together to determine when you sleep and how deeply. But layered on top of that, we now understand what's actually happening during each stage. We know that slow-wave sleep is when the glymphatic system is most active, when growth hormone peaks, when declarative memories are consolidated, when immune memory is strengthened, when synaptic homeostasis does its pruning work. We know that REM sleep is when emotional memories are processed, when procedural learning is refined, when the brain runs through creative associations unconstrained by the logical prefrontal cortex. We know that even the lighter stages of non-REM sleep serve functions. Sleep spindles in stage 2 are correlated with memory integration, and the transition phases help regulate autonomic function. And yet there's still so much we don't know. That's one of the things that makes this field so alive right now. For all the progress, there are fundamental questions that remain genuinely open. Why do we dream? Not just what brain regions are active during dreaming or what neurochemicals are involved, but why does consciousness persist in this bizarre, hallucinatory form during REM? What is the subjective experience of dreaming actually for? Hobson and McCarley's activation synthesis hypothesis from 1977 suggested dreams are essentially noise, random brainstem signals that the cortex tries to weave into a narrative. But that never fully satisfied anyone, and more recent work suggests dreams may serve functions in emotional regulation, threat simulation, even social cognition. Ravonsuo's threat simulation theory proposes that dreaming evolved as a way to rehearse responses to dangerous situations in a safe environment. There's evidence for it, but it's far from settled. The honest answer is that we still don't have a complete theory of why dreams exist, and anyone who tells you otherwise is oversimplifying. There's also the question of individual variation. We touched on Ying Huifu's discovery of the DEC2 mutation, those rare individuals who genuinely thrive on 6 hours or less. But beyond that extreme, there's enormous natural variation in sleep need, sleep timing, sleep architecture. Some people are deep sleepers, others are light. Some people's circadian clocks run slightly longer than 24 hours, others slightly shorter. Morning larks and night owls aren't just lifestyle choices. They're genetically influenced chronotypes, and forcing a true night owl to function on a morning lark's schedule has real cognitive and health consequences. There's growing recognition in the field that sleep medicine needs to become more personalized, more attuned to individual biology, rather than applying one-size-fits-all recommendations. The standard advice of 7 to 9 hours is a population average, and it's good general guidance, but your optimal sleep might be different from your partner's, and that's okay. What matters is whether you're getting enough for your particular biology. And then there's the technology question, which cuts both ways. On one hand, we live in an era of unprecedented sleep disruption. The blue light, the caffeine culture, the always-on work expectations, the screens in every bedroom that we talked about earlier. But on the other hand, technology is also giving us new tools to understand and improve sleep. Consumer sleep trackers, for all their limitations in accuracy compared to polysomnography, have made millions of people aware of their sleep patterns for the first time. Cognitive behavioral therapy for insomnia, which we discussed with Spielman's model, is now available through apps and online programs, making it accessible to people who might never see a sleep specialist. And in the research world, tools like optogenetics and advanced neuroimaging are allowing scientists to manipulate and observe sleep circuits with a precision that would have been unimaginable to Kleitman or Oserinsky. There's something poetic about all of this if you let yourself sit with it. You're lying here right now, and your body is preparing to do something extraordinary. In the minutes or hours ahead, maybe sooner than you think, your brain will begin its nightly descent through the stages of sleep. Your thalamus will start generating those rhythmic waves that gate sensory input, gradually disconnecting you from the outside world. Your prefrontal cortex will quiet down, releasing you from the grip of executive control, from planning and worrying and analyzing. Adenosine, that molecule that's been building up all day, will finally get its way, tipping the balance towards sleep. Your core body temperature will drop, your heart rate will slow, your muscles will relax, and then the real work begins. The glymphatic channels between your brain cells will widen as cerebrospinal fluid pulses through, carrying away the day's metabolic debris. Your hippocampus will begin replaying the day's experiences, sending them up to the cortex for long-term storage. Synapses that were strengthened during the day will be carefully recalibrated, the important ones preserved, the trivial ones pruned back, restoring your brain's capacity to learn tomorrow. Your immune system will take stock of every pathogen it encountered and begin writing those encounters into lasting memory. Growth hormone will surge, repairing tissue, building muscle, maintaining bone. All of this happens without you doing a thing. You don't have to direct it. You don't have to understand it. You just have to let it happen. And that might be the most remarkable thing about sleep, that this incredibly sophisticated, multi-system, precisely orchestrated biological process requires nothing from you except surrender. Just letting go. Just allowing your brain to do what it has evolved over hundreds of millions of years to do. Because sleep isn't a modern invention. and it isn't a human invention. Every animal with a nervous system sleeps in some form. Fruit flies sleep, jellyfish sleep, or at least they enter a state that meets the behavioral criteria for sleep, which is a wild thing to contemplate given that they don't even have a brain. Sleep is so fundamental, so deeply embedded in biology, that it predates brains themselves. Whatever sleep does, it's so important that evolution has preserved it across every branch of the animal kingdom, despite the enormous vulnerability it creates. An animal that sleeps is an animal that can be eaten. And yet, sleep persists. That tells you something about how essential it must be. So as you lie here, feeling the weight of your body settling into the mattress, feeling your breathing slow and your thoughts begin to soften around the edges, know that you're about to join a process that connects you to every sleeping creature on this planet. The sparrow tucked into a hedge outside your window, sleeping with one eye open and half its brain awake. The dolphin drifting in warm water, sleeping one hemisphere at a time. The bear in its den, the cat curled on someone's couch, the fruit fly clinging to the side of a glass somewhere in a lab. All of them cycling through their own versions of this ancient, necessary process. You don't need to think about any of this anymore. The science will be here tomorrow. The history, the researchers, the discoveries, they'll all be waiting. Right now, the only thing your brain wants is permission to begin its work. And the beautiful thing is, it already knows how. It's been doing this every night of your life, long before you knew what adenosine was, or what a sleep spindle looked like, or why your body temperature drops before sleep onset. Your brain has been running this program flawlessly, night after night, without any instruction from you at all. So let it. Let the warmth of the bed hold you. Let your thoughts drift where they want to drift. If they circle back to something we talked about, Michel Cifre in his cave, or Colin Sullivan tinkering with a vacuum cleaner motor in a Sydney hospital, or Mike in Nedergaard watching dye flow through the sleeping brains of mice, that's fine. Let those images float by like clouds. And if your thoughts dissolve into nothing at all, into that soft, formless space where waking ends and sleep begins, that's fine too. That's exactly where you're supposed to be. Your brain will take it from here.